My name is Tolstoganov, and I'm going to present BinSPreader, which is a tool for refining metagenome assembled genomes. It is, of course, a joint work with Yuri Kamenev, Roman Kruglikov, and Anton Korobeynikov. Metagenome assembled genome, or MAG, which we try to reconstruct, is a set of contigs from a metagenomic assembly, which allegedly represents a group of closely related species. The common problem with MAGs constructed by state-of-the-art tools is that they're often incomplete or contaminated, especially for complex metagenomic samples. Conventional MAG reconstruction workflow looks like this. First, we obtain a whole genome shotgun sequencing sample or series of samples of microbes from their host environment, generate a combined assembly of the sample, and split the contigs from this assembly into bins using information such as coverage profiles or TNF distance. Then these preliminary bins are somehow refined to produce more accurate and complete MAGs. In reality, modern workflows such as MetaRep are considerably more complex, and bin refinement modules in these workflows are based on combining results from multiple binning tools. Unfortunately, these consensus-based refining tools do not solve one of the main problems of MAG reconstruction, which is that almost all binning tools remove short contigs to filter out noise from the data, and as a result, a large part of the assembly is missing from the bins. And to make matters worse, these missing short contigs often represent important stuff, such as conservative genes and strain variants. So consensus approach does not help us to include short contigs into bins, since they are ignored by individual binning tools. Existing bin refinement approaches can be divided into three groups: consensus-based approaches, such as MetaRep and DASTool, that combine results from multiple binners, Binnacle, that utilizes paired end information, and GraphBin2, which utilizes scaffold graph to propagate binning from long contigs to short contigs. Our method for binning refinement is called BinSPreader, and we also concentrate on propagating bins to short contigs. In addition, our tool is also able to correct binning errors, assign multiple bins to a single contig, split reads according to bins in the assembly graph, and calculate the distances between bins based on the overlaps between them, in order to estimate the phylogenetic distances between the corresponding genomes. Since we're using assembly graph instead of consensus information, our method can be used to further improve the results of consensus-based refiners. One of the main differences between our tool and GraphBin2 is that we use the assembly graph instead of the scaffold graph, because the assembly graph usually gives more accurate representation of the contig connectivity information. For example, say that we have a common situation in the assembly graph, a partially unresolved intergenomic repeat, where we correctly matched one of the entrances to one of the exits and merged them into a single contig along with the repeat edge, but we did not resolve the other entrance-exit pair. But based on some information other than connectivity, for example, coverage, we were able to assign these two edges into a red bin. So as a result, we have 5 edges, 2 contigs, 3 contigs, and 2 bins. If we use a scaffold graph representation of the assembly graph, we might try to correct the blue contig based on the graph information and reassign it to the red bin, which is, of course, a false correction. But if we use edge adjacency graph, where vertices represent assembly graph edges and edges represent shared vertices between assembly graph edges, we have more accurate representation and are able to assign both red and blue bin to the repetitive edge without making any false corrections. So how do we actually use assembly graph to refine bins? Well, first, let's just formulate a more clear description of a bin refining problem. What we want to obtain is a soft bin matrix F, where element Fij is a probability of assigning bin j to edge ei of the assembly graph. And what we have is the initial binning matrix Y, where element Yij is 1 if bin j is assigned to edge ei and 0 otherwise. Since initial binning usually misses short contigs, Y has a lot of 0 rows, which is not what we want from F, but we mostly trust the initial assignment for long contigs. So our preferred result is a soft bin assignment matrix, which is close to I, but does not contain 0 rows, since we want to propagate bin labels from long contigs to short contigs. So what do we mean by close? By close, we mean that we try to minimize the following cost function, where the first term controls the bin label smoothness along the assembly graph, because we assume that two edges that share a vertex in the assembly graph probably have similar bin labels. And the second term penalizes the distance between resultant binning and original binning, according to regularization parameters defined separately for every edge. We can choose different regularization parameters for different kind of edges and for different modes of BinSPreader. For instance, if we assume that the initial binning is accurate, we do not want to change the initial binning at all and set the regularization parameters to infinity. Conversely, if we have short edges that belong to multiple scaffolds, we know that they're definitely repetitive. So assigning a single initial bin to them does not make any sense, so we completely ignore the initial binning. In general, all short edges are likely repetitive, so for them, we can optionally use a low regularization parameter, such as logarithm of length. So by minimizing the cost function, we obtain soft bin assignments for edges, and now we need to get bin assignment for scaffolds by summing bin likelihoods of edges of the scaffold based on length and soft bin labels on the edges. Then we use these likelihoods to obtain either single or multiple scaffold bin assignment, depending on the edge multiplicity and whether we allow the multiple bin assignment for long edges or not. We can further utilize those refined bins with multiple assignment for various purposes. For example, if we have raw reads instead of just the assembly, we align them back down to the assembly graph, split reads according to the bin assignment, and reassemble them with individual bins to improve the quality of the assembly. We also try to estimate the phylogenetic distance between species represented by our bins. If we again return from bin labels on scaffold to bin labels on edges, we can consider our bins as a probability distributions on the assembly graph edges. So we can measure similarity between a pair of bins using probability Jaccard index JP and use 1 minus JP as a bin distance estimation. If the distance between two bins is low enough, we can assume that they actually represent the same species and merge them into a single bin, further improving the quality of our binning. So moving on to the results, we benchmarked our tool on 3 synthetic datasets, which are datasets from the non-mock communities cultivated in the lab with known ground truth. So these 3 datasets, Synth, Embark, and BMOC12, consist of 64, 26, and 12 species respectively. Also, BMOC12, despite its small size, is actually quite complex since it contains 5 closely related species. To demonstrate this, here is our bin distance estimation for BMOC12 and Embark. Nodes represent initial metabad bins that we refine. Node color represent the most abundant genome in the bin, and the tree is built using hierarchy clustering from our estimated distance. So we can see that bins from 2 closely related Marinobacter species in BMOC12 are mixed in a single branch. And the same happens on Embark, but to a lesser extent. To benchmark the actual refining, we compared BinSPreader with GraphBin2, which is a scaffold graph-based tool. And both BinSPreader and GraphBin2 used MetaBat2 for initial binning. So this table shows the number of recovered mags with given contamination and completeness. Contamination here is the fraction of mag that doesn't come from the most abundant genome, and completeness is the fraction of the most abundant genome present in our mag. So BinSPreader reconstructed 22 out of 25 known genomes with less than 10% contamination and 20 with less than 5% contamination. And when we look on BMOC12, BinSPreader reconstructs only 5 genomes with 90% completeness due to the presence of closely related organisms, but it's still more than the initial MetaBat2 binning and than GraphBin2. And perhaps the reason for this is that refining tools may actually increase the contamination on complex dataset. So here are purity and completeness plots for individual bins, which also consider their length. Basically, what happens on the left plot is that we sort bins in the descending order of completeness, and x coordinate on the nth point on the plot corresponds to the total length of the first n sorted bins, while y coordinate on the nth point corresponds to the completeness of the nth bin. On the right side, there is the same plot for the purity. So we see that both GraphBin2 and BinSPreader significantly increase the completeness of the initial bins, but BinSPreader does not introduce as many errors. The results are similar for embarked dataset, except that in the absence of closely related organisms, a BinSPreader does not introduce any contamination. So a large motivation to include short contigs into MACs is the reconstruction of conservative genes, such as 16A RNase genes. And we can see good results on all three synthetic datasets. Again, a bit worse on BMOC12 because of closely related species. So currently we are using only assembly graph information for binary refinement, but basically the same algorithm can be adapted to different types of genomic data, such as the ordinary paired dendrites, IC, long reads, read clouds, and so on, which sets ground for the future work. At the moment, BinSPreader exists only as a part of a developer version of Spades, unfortunately, so please contact us by the Spades support email for an early access version. I would also like to thank my co-workers at the Center for Algorithmic Biology for many useful ideas and suggestions, and the Russian Science Foundation for the financial support of this work. Thank you very much for your time.